In this video presentation, we will discuss about rabies, one of the most important zoonotic viral disease in dogs, under the Rhabdoviridae viral family. Other important viral disease infecting dogs apart from rabies are canine parvovirus and canine distemper. In this lecture, we will discuss in detail on rabies infection. Classification: Rhabdoviridae family classified under the Baltimore Group 5 with the order Mononegaviral. Group 5 comprises of viruses with single-stranded negative sense RNA genome. In the word mononegaviral, mono means the monopartite, that is single-segmented genome, and nega means the negative sense RNA genome. Some of the animal important viral diseases under this family are rabies under the genus Lyssavirus, bovine ephemeral fever under the genus Ephemeravirus, and vesicular stomatitis under the genus Vesiculovirus. In this lecture, we will discuss in detail on rabies infection. Rabies. The other names for this infection are hydrophobia and mad dog disease. The word rab is derived from Sanskrit, means to do violence. The disease is caused by the genus Lyssavirus. Based on tissue tropism, this virus is having target towards the neuronal tissues, so they are termed as neurotropic virus. This virus causes fatal encephalitis in humans and in many mammals. Host range. This neurotropic virus causes infection in dogs, cats, wild cats, jackal, wolf, bat and humans. This infection can be transmitted from animals to human, so they are considered as zoonotic infection. This virus is classified as risk group 3 pathogen. Direct human-to-human transmission is theoretically possible, but rare, and has only been documented in cases of organ transplantation. Pathogens can be classified based on their risk to humans, namely risk group 1, risk group 2, risk group 3 and risk group 4. The agent which does not causes infection in human, and the treatment is not required, are classified as risk group 1 pathogens. The agent which causes mild infection in human, and the treatment is available, are classified as risk group 2 pathogens. The agent which causes serious infection in human, and the treatment is available, are classified as risk group 3 pathogens. And the agent which causes serious infection in human, and the treatment is not available, are classified as risk group 4 pathogens. So, in rabies, the virus causes serious infection in human, and the treatment is available. So, they are grouped under risk group 3 pathogen, and must be handled only in laboratories with biosafety containment level 3 facilities. History. In 1885, Louis Pasteur gave the first human vaccination against rabies, to a boy named Joseph Meister, who has been bitten severely by a rabid dog. This period was considered as era of infectious disease prevention. Later, in 1903, Remelinger and Riffet discovered the causative agent, the rabies virus. Virus morphology. The virus possesses helical capsid, which is about 180 nanometer diameter. The virus possesses envelope and glycoprotein spikes. There is also an additional protein, the matrix protein, which play a major role to maintain this bullet-shaped organization. Imagine, if this matrix protein is removed, the entire structure becomes highly extended and filamentous. The illustration showing the viral morphology in detail. The virus possesses helical capsid, with the matrix protein over it, and enveloped. The entire virus structure is about 180 nanometer size, which is studded all around with the glycoprotein spikes. Among all, the glycoprotein spike is the immunodominant protein, which play a major role in the antigenicity. Genomic organization. The genome is single-stranded, monopartite and negative sense RNA. So the virus is under the order Mononegavirales. The genome length is 11 kilobase pair length. The viral genome encodes 5 proteins, namely, N gene, which codes for nucleoprotein, P gene, which codes for phosphoprotein, M gene, which codes for matrix protein, G gene, which codes for glycoprotein, and L gene, which codes for polymerase enzyme. The diagnostic tests mainly targets for nucleoprotein, that is N gene for identifying the viral antigen. As we already discussed, among all the protein, the glycoprotein is the immunodominant protein, which play a major role in the antigenicity, and helps in the development of new generation vaccine. Here, the genome is negative sense RNA, that is, they are complementary to mRNA. So, they cannot translate, or be infectious on its own. So first they must be converted to positive sense RNA, by using virus-encoded transcriptase enzyme, that is, RNA-dependent RNA polymerase. Then, This transcribed positive sense RNA, which are similar to mRNA, can be translated into viral proteins. Virus replication. The rabies virus replicates in the cytoplasm. First, the negative sense viral RNA are converted to positive sense by using virus-encoded transcriptase enzyme, that is, RNA-dependent RNA polymerase. Then, this transcribed positive sense RNA, which are similar to mRNA, is translated into viral proteins. On the other side, the negative sense viral RNA are transcribed for complementary strand, for the transcription of negative sense viral RNA. Later, this transcribed negative sense viral RNA, and the translated for viral protein synthesis, self-assembles to form virion. Distribution. This viral infection is endemic for many countries. But, places like United Kingdom, Norway, Iceland, Australia, New Zealand, and Japan are listed as rabies-free countries. Among the rabies death rate across the globe, India accounts for 36% cases. Among the rabies death rate across the Asia, India accounts for 65% cases. Genetic lineages. There are also other viruses listed under the genus Lyssavirus, like Macola virus, Duvenhaga virus and Lagos bat virus from Africa. Then European bat Lassa virus, which possesses two biotypes, and Australian bat Lassa virus. These all viruses are considered as distinct genetic lineages with rabies virus. Physical inactivation. The rabies virus get inactivated at the temperature of 50 degrees Celsius, for 1 hour, or, at the temperature of 60 degrees Celsius, for 5 minutes. These viruses are also sensitive at the pH less than 3 or at the pH more than 11. The ultraviolet light inactivate this virus. This virus does not survive well outside its host, as it is susceptible to sunlight and desiccation. Chemical inactivation. The rabies virus is inactivated by exposure to 70% ethanol, phenol, formalin, iodine, trypsin, beta-propiolactone, soaps, detergents, and lipid solvents, such as ether, chloroform, and acetone. Antigenic properties. The rabies virus possess surface spikes, composed of glycoprotein G. The surface glycoprotein G spikes binds to acetylcholine receptors in the neural tissue. In rabies, two types of life cycles exist. One is sylvatic, or jungle cycle, and the other is urban cycle. In the sylvatic cycle, transmissions are existing between wildlife such as fox, coyotes, wolf, skunk, raccoons, hyena, and bats. In urban cycle, transmissions are existing animals such as dogs, cats, monkey, horse and cattle. So, the human can get the infection, either through sylvatic cycle, or through urban cycle. But, humans are highly prone from getting infected through urban cycle. Source of infection. Infected animal, that is, rabid animal, act as the principal source of this virus. Transmission is through saliva, brain materials, bites of rabid animal, and licks of rabid animal, through an open wound, through mouth, eyes, and through mucous membranes of nose. Pathogenesis. The virus first enters the tissue, from saliva of biting animal. Then, the virus do its initial replication, in the muscle tissue near the bite. Following replication, the virus binds to the acetylcholine receptors in the nearby nerves, with the help of surface glycoprotein G spikes, which we have discussed earlier. Then, the virus moves up through peripheral nervous system to central nervous system. Once the virus enters the central nervous system, it ascends through spinal cord, to reach the brain, where it causes fatal encephalitis. After its replication in the brain, the virus moves to salivary gland, and other organs. So at first, the virus is moving from outside, that is, from muscle, toward the brain, that is, towards the center. So there is a centripetal movement is followed. Then, the virus is moving away from center, that is, away from brain, to the salivary gland, that is outside. So, there is a centrifugal movement is followed later. Pathogenesis. The virus first enters the skin tissues, from saliva of biting animal. Following initial replication, the virus moves up through peripheral nervous system to central nervous system. So there is a centripetal movement is followed initially. Then the virus moves from central nervous to salivary gland. So, there is a centrifugal movement is followed later. Incubation period. The incubation is about 14 to 90 days, or more. The period varies. It depends upon, number 1. Site of rabid dog bite. Whether the bite is near to head, or far from head. Whether the bite is at extremities, like fingers, where the nerve endings are abundant. Number 2. Number of bites. Whether it is single or multiple rabid dog bites. Number 3. The amount of virus inoculated during bite. More the viral load, shorter the incubation period. Clinical manifestations. Different clinical manifestations are observed in different stages of rabies. Starting up with prodrome phase. Then, acute neurologic encephalitis, that is furious form. Paralysis followed by coma, that is dumb form. And then death. But in rare case, the recovery may be observed. In prodrome phase, fever, headache, depression, and upper respiratory and gastrointestinal tract disorders observed. This prodromal phase typically lasts about 4 days, but can last as long as 10 days, before specific symptoms develop. Furious, or encephalitic form. In this stage, the animal will be restless, nervous, aggressive, dangerous, loses fear of humans, bites at anything. In this stage, hydrophobia, that is, the animal cannot swallow the water, is observed. Other signs like excessive salivation, exaggeration to light and sound, and the animal will be hyperesthetic is observed. This fury form, most commonly observed in dogs, cats, and horses. The next stage is dumb or paralytic form. Following furious form, the animal will be paralyzed. Following paralysis, coma, and death is noticed, due to respiratory arrest. In cattle, in other ruminants, and in lab animal, only dumb form is observed. On post-mortem examination of brain, encephalomyelitis, that is inflammation in brain and meninges, is observed. Some of the findings are mononuclear infiltration, paravascular cuffing of lymphocytes or polymorphonuclear cells, and presence of NEGRI bodies are noticed, microscopically. The picture at the right, showing the microscopic finding, perivascular cuffing in brain tissue section. NEGRI bodies are the viral inclusion bodies. Here, the virus replicates inside the cytoplasm, and form an aggregate of virus particle within the cytoplasm of nerve cells. These inclusion bodies are round, or oval, inside the cytoplasm. This inclusion can be stained by Mann's or Seller's method. These methods helps to differentiate rabies inclusions from other intracellular inclusions. The electron microscopic picture at the right, depicting the intracytoplasmic viral inclusion bodies in the neuron of a rabies case. For demonstration of NEGRI bodies, in the unfixed tissue sections, Seller's staining method is followed. For demonstration of NEGRI bodies, in the fixed tissue sections, Mann's staining method is followed. The rabies virus can be street virus or fixed virus. The street virus are the virus recovered from naturally occurring rabies cases. But the fixed virus are recovered as a result of repeated culture in rabbit brain, which has short, fixed, and reproducible incubation period. The street virus can produce NEGRI bodies. But the fixed virus cannot form NEGRI bodies. The street virus have long incubation period, whereas the fixed virus have short incubation period. The street virus is pathogenic for all the mammals. But the fixed virus is pathogenic under certain conditions to humans. The street virus cannot be used for vaccine preparation, whereas the fixed virus can be used for vaccine preparation. Diagnosis can be done in field level or in the laboratories. Field diagnosis. Based on the clinical symptoms, such as furious and dumb form, as we discussed earlier. Next. Laboratory diagnosis. For lab diagnosis, cerebrospinal fluid, saliva, serum and nuchal skin biopsy are collected from live individual. In dead, brain tissue is collected. In brain, these viruses are abundant in thalamus, pons, and medulla. For lab diagnosis, sample has to be sent for diagnosis to Neurovirology Lab at National Institute of Mental Health and Neurosciences, NIMHANS, Bangalore. Disease Investigation Lab at Directorate on Animal Husbandry and Veterinary Services, Goa. IIMS, Jodhpur. National Center for Disease Control NCDC, Delhi. And Rabies Diagnostic Lab, at Veterinary College, Bangalore. This virus can be cultivated or isolated in lab by 3 ways. Number 1. Animal inoculation. Here, the newborn mice is inoculated with the virus through intracerebral route inoculation. After inoculation and incubation, the mice develops paralysis, and the death will be noticed within 28 days. Then, the presence of rabies virus can be confirmed by examining the mice brain using direct fluorescent antibody technique. This method is called Hobble mouse inoculation test. Hobble is the scientist who has devised this inoculation test. The second method of cultivation of this virus is cell culture system. Here, cell lines like WI38 cells, BHK21 cells, that is baby hamster kidney cells, and mouse neuroblastoma cells can be used. This virus cause minimal cytopathic effect CPE over the cells. The third method of cultivation of this virus is embryonated chicken egg. Allantoic cavity inoculation on 9 to 11-day chicken embryos can be done. Here, the lesions caused by the virus over embryo are irregular, and not characteristic. These are some of the laboratory tests can be done for diagnosis of this virus. For antigen detection, direct fluorescent antibody test, immunoperoxidase test, enzyme-linked immunosorbent assay ELISA, reverse transcriptase PCR by targeting the N gene, DNA sequencing, and electron microscopy can be done. Direct fluorescent antibody test. Most commonly used test for rabies diagnosis. This test is considered as a gold standard test, recommended by WHO and OIE. In this test, fluorescein, isothiocyanate conjugated, that is FITC conjugated anti-rabies antibody is used. This labeled detecting antibody is used to detect the rabies viral antigen in the test sample, or in the tissue sections under ultraviolet microscopy. For antibody detection, RFFIT, rapid fluorescent focus inhibition test, is done to evaluate the rabies virus neutralizing antibody titer in the serum and cerebrospinal fluid. This test, in absence of vaccination history, offers an indirect way to evidence the rabies infection. Prevention and control. Before going into the preventive and control measures, we will discuss in detail about the rabies vaccines used over different years. These are the rabies vaccines used over different timelines, starting with the first, the Pasteur-type vaccine. Other nervous tissue vaccines like FERMI and SEMPLE vaccine. Avianized vaccines. Duck embryo vaccine. Human diploid cell vaccine. And other cell culture vaccine. Pasteur-type vaccine. This is a nervous tissue vaccine, oldest vaccine. Developed by Louis Pasteur in 1885. Not in use at present. Here the fixed virus was injected into the rabbit. Then, the rabbit brain cords are collected, and dried. Here the virus inactivation was done by drying the nerve tissues. FERMI vaccine. Developed in 1930s. It is also a nervous tissue vaccine, where the virus is injected into rabbit, and the infected rabbit brain is used. A modification here is, phenol is used for virus inactivation. SEMPLE vaccine. Developed in 1930s. It is also a nervous tissue vaccine. A modification here is, the virus is injected into sheep, and the infected sheep brain is used. The virus inactivation is with phenol. But these nervous tissue vaccines are associated with the rare complication of demyelinating allergic encephalitis. 4 cases for 1,000 persons vaccinated, developed this encephalitis complication. Next is avianized vaccines. Developed in 1940s. Here Flurry vaccine strain is grown in embryonated chicken egg. 2 types are there. First, low egg passage, LEP vaccine, where 40 to 50 viral passages in embryos are done. Following passages, it loses its pathogenicity for rabbits and dogs. This vaccine is used only for dogs, and not for any other animals. Second type is high egg passage, HEP vaccine, where 180 viral passages in embryos are done. Following passages, it loses its pathogenicity completely, except for mice. This vaccine were used for humans also. Duck embryo vaccine. Developed in 1950s. Here, the virus is cultivated in embryonated duck eggs, followed by virus inactivation using beta-propiolactone. The advantage of these embryonated vaccines over nervous tissue vaccines are lower risk of allergic encephalitis. But they are less immunogenic, and with minor side effects like fever, depression, myalgia, and generalized lymphadenopathy. Human diploid cell vaccine. Developed in 1970s. Here, the virus is cultivated in human diploid cells like MRC5 cells or WI38 cells. These cells are derived from normal human fetal lung fibroblast tissue. Here the virus is cultivated in human diploid cells, followed by virus inactivation using beta-propiolactone is done. These vaccines are highly effective, and adverse reactions are extremely rare. But they are very expensive, due to use of human cell culture. Last, the other cell culture vaccine. Developed in 1980s. These vaccines are newer and less expensive vaccine. Here the virus is cultivated in the chicken embryo fibroblast cells, or in cells. Followed by virus cultivation, inactivation of virus using beta-propiolactone is done. Vaccines under this types are purified chicken embryo cell rabies vaccine, PCECV, and purified varicella rabies vaccine, PVRV. In India, where rabies is endemic, 4 types of rabies vaccines are available. The first 3 are the cell culture derived. Number 1, human diploid cell culture vaccine. Number 2, purified chick embryo cell vaccine. Third, purified varicella rabies vaccine. And the fourth is the purified duck embryo vaccine. These are the rabies vaccine strains: PV11 strain, Pittman-Moore strain, Fleury strain, Wistar strain, and CVS strain. Molecular studies have shown that these strains are genetically homologous. Prevention and control. 2 different regimens are followed. One is for pre-exposure prophylaxis, that is, before dog bite or before exposure to the virus. And another is for post-exposure prophylaxis, that is, after dog bite or after exposure to the virus. For pre-exposure prophylaxis in humans, the vaccination using anti-rabies vaccine ARV is followed. This measure is important for veterinarians, animal handlers, lab workers employed in the rabies laboratories, and traveler or geographic explorers traveling to the forest, caves and rabies endemic areas. For this, ARV is given at the dose of 1 ml intramuscularly into the deltoid muscle, or 0.1 ml intradermal injection is given. Intradermal injection is more economic and antigen-saving alternative to intramuscular administration, as it requires 0.1 ml of vaccine per intradermal injection dose, in contrast to 1 ml per intramuscular dose. ARV administration schedule is followed on 0 day, 7th day, 21st day or 28th day. Revaccination is followed every 3 to 5 years, that depends upon the neutralizing antibody level. Antibody levels can be evaluated by rapid fluorescent focus inhibition test RFFIT, which we discussed earlier in lab test. For pre-exposure prophylaxis in animals like dog and cat, the ARV vaccination is done as per VSAVA guideline. That is the global guideline given by World Small Animal Veterinary Association. Vaccination schedule. The primary vaccination in the dogs and cats against rabies is done at 3rd month of age, that is 12th week, with the dose of 1 ml per animal, intramuscular. Followed by 2 to 4 weeks apart, next vaccination at 4th month of age, that is 15th week is done. In order to maintain the protective immunity against rabies, revaccination is done, every year, that is annually. Oral vaccination against rabies. This is done to prevent and eradicate rabies in wild animals, such as foxes, raccoons, coyotes, and jackals. Here, the vaccine is mixed with the edible food, and used as bait vaccine. This bait vaccines are distributed by airplanes in wild and rural areas. The left picture, showing the paraffin-coated edible food bait vaccine. The right one, showing the dog, consuming the bait vaccine. For post-exposure prophylaxis, that is after suspected rabid dog bite, following PEP measure are followed. Number 1. Wound management. Number 2. Vaccination. And number 3. Rabies immunoglobulin, that is RIG infiltration. We will discuss in detail about each measures here. Following wound management measures is to be followed immediately. Washing the wound with soap and water. Then, disinfecting the wound with disinfectants like alcohol, povidone iodine and Dettol. The wound should be kept open, for exposure to sunlight. Suturing of wound should not be done. And the local irritants should not be applied over the wound. TT injection and antibiotic also administered to combat secondary bacterial infection. These are the basic and foremost important regimen to be followed after dog bite. Next, vaccination, that is active immunization. Here, anti-rabies vaccine is given at the dose of 1 ml intramuscular, or 0.1 ml intradermal injection. The vaccination is followed on 0 day, 3rd day, 7th day, 14th day and 28th day. The 90th day vaccination is optional. Once an individual is immunized with rabies vaccine, that does not mean he is protected against rabies lifelong. Third, RIG infiltration. Rabies immunoglobulin infiltration. Also called as anti-rabies serum. This is recommended even a microdroplet of blood is noticed. Here the infiltration of rabies immunoglobulin is done into and around the wound on 0 day or within 7th day post-bite. Once infiltrated, the rabies immunoglobulin will bind to the virus, and neutralize it at the site of bite. RIG preparations like equine rabies immunoglobulin ERIG and Human rabies immunoglobulin HRIG are available. ERIG at the dose rate of 40 international unit per kg body weight, and HRIG at the dose rate of 20 international unit per kg body weight is infiltrated around the wound on 0 day or within 7th day post-bite. Following are the summary about the PEP regimen to be followed. Wound management. Immediate and thorough wound washing, with soap and water and disinfections. TT and antibiotic administration. Anti-rabies vaccination on 0, 3, 7, 14 and 28th day with 90th day optional. And rig infiltration around the wound. The contact with suspect rabid animal is classified into 3 categories. Category 1. Licks on intact skin. Category 2. Nibbling of uncovered skin, minor scratches or abrasions without bleeding. And Category 3. Nibbling of uncovered skin, minor scratches or abrasions without bleeding. Single or multiple transdermal bites or scratches, licks on broken skin, contamination of mucous membrane with saliva from licks, contacts with bats. Here are the categories of contact and their recommended post-exposure prophylaxis measures to be followed as given. For Category 1 contact, washing is mandatory. For Category 2, washing plus vaccination is must. And for Category 3 contact, washing, vaccination and rig infiltration is followed. September 28th. World Rabies Day. This day is celebrated for Dr. Louis Pasteur's vision of rabies-free world. Rabies Day is a cooperative global event planned to reduce the suffering from rabies worldwide. Hope the lecture is informative and useful. Thank you.